to begin with Dr. David Wong. Dr. Wong is a distinguished professor from the University of California, Los Angeles. He is also director for the Eye Center for Oral, Head, and Neck Oncology Research. And his presentation today is going to be Salivaomics, Exosomics, Saliva Liquid Biopsy. Dr. Wong, over to you.
Thank you, Brittany. I'm just going to bring up my slides. And is it coming up? Whoops. There we go. Brittany, you see my slide coming up?
Yes, your slides are up.
Okay, very good. So thanks, Brittany, and want to thank Saliva Direct for having me here. Looking forward to spend time with you, to share with you this portfolio that we're all excited about. So I'll just I can advance it from here. My talk is Salivaomics, Exosomics, and liquid biopsy. And the following slide is my disclosure slide. And so I'll advance forward from here. So if and when saliva, this is oral fluid, mature towards clinical, deployment, there needs to be two frontiers that needs to advance and eventually converge. And one is the translational frontier, where there needs to be developed biomarkers that are definitively and pivotally validated, and I think the point is will be clear throughout these webinars. Perhaps the other and equally important is a scientific underpinning. How, particularly when systemic disease are being conveyed and reflected in saliva, and incredibly developed and then clinically deployed. So these mechanistic underpinning is vitally important. The topic perhaps that I would like to share with you in particular, as we look at the saliva as a body fluid that could convey this capability or utility, is these omics constituents that are endowed within this fluid that are present in other body fluid that are so germane in personalized medicine and in precision medicines. That perhaps the three major omics constituents I want to cover today is transcriptomics, proteomics, and genomics. So these endeavors actually have been in development and maturation for the past perhaps decade and a half. The proteome, as many of us know, that our last speaker, I think, is our last speaker is Marcello Fiore at the Quake Venture Institute, is actually currently hosting this initial initiative that were embarked upon by the Dental and Craniofacial Institute, and we're a part of that. And the other its transcriptomic constituents was recently, in the past decade, undertaken by the Common Fund to comprehensively profile and decipher all the extracellular RNA body fluid. And saliva is part of that constituent outcome that I'll highlight to you later in my talk. And perhaps what importantly is, as these omics constituents reflect itself in this body fluid, its utility continue to emerge to most recently, as you all know, and particularly the impact that Saliva Direct is towards reflecting the genomic RNA of SARS-CoV-2 in this oral fluid. And we sort of have seen that clearly, impactfully, but also the ability to utilize these other omics constituents that in our effort towards this pandemic is to address, assess, and perhaps communicate 4-dimension of this pathogen's information in a drop of this oral fluid: genomics, antigenically, immunoglobulin, and neutralizing antibody. So these excitement really emerge from these omics constituents in their development. In the transcriptomics sector, the initial discovery, if you will, or footprint of an extracellular RNA in saliva was almost 2 2 decades ago, when we first published that, using at that time a microarray technology, a transcriptomic profile in saliva of healthy individuals, and then eventually demonstrate its utility in detecting oral cavity cancer. And that capability has further advanced into an ability to benchmark against these extracellular RNA in a seromal compartment. And perhaps most gratifying is that, even as we reported 2 decades ago, it was met with a lot of skepticism, and rightfully so, perhaps, because the existence of RNA in a quote unquote a hostile fluid, where actually the degradation enzyme RNase is present in saliva, could actually convey these clinical utility. And this activity was the common fund of the NIH 10 years ago. Set up an extracellular RNA consortium where they invest $400 million to comprehensively decipher, profile, catalog, and annotate the entire extracellular RNA body fluid, plasma, serum, cerebrospinal fluid, and we were in that activity of cataloging and extracellular RNA in saliva. So this initiative was important because it not only is ability to profile and catalog these extracellular RNA in bodily fluid, but the existence of these RNA as an extracellular constituents that are either in sort of naked form, these wiggly molecules, I'm trying to get my cursor on screen here, or are they encapsulated in these extracellular vesicles? Okay, so this is a new paradigm where RNA could exist outside a cell, from a cell of origin, this neuron here, and into a columnar epithelial cells, where they could be intake and translate into protein that would influence the physiology of that target cell. So we were in this consortium for 2 cycles, and this existence translate into an opportunity to decipher, if you will, the entire transcriptomic content in saliva. Our first work was done with life technology, where we deployed RNA-seq for the first time in saliva, and this is that particular outcome. And perhaps the one feature I want to highlight is the most highly expressed content within this fluid are non-coding RNA in both host saliva and cell-free saliva. This was we're caught by surprise, and so to speak. We went further to comprehensively decipher the non-coding RNA in saliva. Worked with Grace Schall in integrated biology, and Dr. Family in my lab. We comprehensively decipher the extracellular RNA, the non-coding sequences, and that was a very important endeavor as it decipher these non-coding elements that are highly expressed in this body fluid. So this is the extracellular RNA atlas. It's in the public domain. Invite you all to take a look at it, and what is being showcased here is all the body fluid, if you focus on this biofluid here, that are being surveyed, if you will. We see serum, urine, cerebrospinal fluid, and we see this beautiful green sector of 246 RNA sequences, long and small RNA, that's being part of this definitive foundation, if you will, of the extracellular RNA. So that's gratifying, and all these concerns and perhaps is now the existence or utility, if you will, of RNA in saliva is no longer being challenged. I mean, again, COVID is clearly a stellar example of that. So with that in mind, this is again in the public domain. We can all go and look at that website. The existence of these constituents allow us to begin to address questions that are of importance in saliva diagnostic. Mechanistically, how does systemic disease, when it developed, that could be conveyed and traverse through a body fluid into salivary gland, into saliva? So we put that to evaluation here. This is a body of work that almost is a decade and a half ago, where animal model were developed pancreatic cancer in rodent. We harvested tumor, the blood salivary gland and saliva, and at that time used microarray to track, if you will, these organ-specific RNA through vasculature, through salivary gland into saliva, and indeed there was a panel of RNA that are disease specific that are being conveyed to, from the organ of pathology into salivary gland and saliva. And furthermore, this study also addressed the vehicle, if you will, the mechanisms that convey these targets from the organ of pathology through vasculature into saliva. And EV at that time is fairly sort of a novel concept. And one of our postdoc fellow, Michael Lau, put that to test and evaluate if he were to knock down the biogenesis of EVs by knocking down RAP11B, if you will, and he indeed observed the ablation of these RNA that present itself in saliva. And it was a very important moment, because this is really the first demonstration of a mechanistic underpinning how a systemic disease could convey itself, traverse through body fluid, and find its way into salivary gland. Is like that vehicle. Is it a Chevrolet? Is it a Toyota that convey these passengers from an organ of pathology into salivary gland, into saliva? So it really presents itself importantly, because that question is going to be asked. How does it get there? Particularly when it's systemic background. So this was very gratifying. It's the second cycle of the Common Fund initiative give us further, footprint into the foundationing of salivary diagnostic, and that is we advance a technology where we can look at single EVs, single exosomes, microvesicles that could traverse these extracellular RNA. This is a panel here for gastric cancer in the case, which is our model, and from the organ of pathology through vasculature to the salivary gland into saliva, and that outcome is really quite astonishing. So I think it will become clear as these seminar, not just today, that the eventual utility, the moment perhaps, not only are we excited about how saliva could be of clinical value and utilities, is perhaps comparative, value and performance over other body compartments will emerge, fairly soon, if you will. So I'm going to advance my slide here. The second domain I want to discuss with you is the proteome, and it was an initiative again through a NIH initiative where 3 groups came together to comprehensively decipher, catalog, and annotate the salivary proteome. So this is our group at UCLA. We work with Julian Whitelake, Joe Liu, and Kim Fo, and also with Susan Fisher's group at UCSF and John Yates at Scripps. So collectively, the 3 groups published the first blueprint, if you will, of the saliva proteome. And what we're looking at here is that of the 1,166 proteins that were sort of identified, 2/3 are in the parotid fluid, and about 1/3 is in the submandibular sublingual fluid. And this Venn diagram actually informs us about 2/3 of proteins are common, and yet 1/3 of proteins are uniquely present in the submandibular fluid and in the sublingual fluid. This is factual, but it's important as we look down the road of a specific proteomic target as it emerges itself in saliva, which glandular compartment would it be coming from from those perspectives. But so this as we heard from Dr. Machello last time, that the salivary proteome outcome has been cataloged and mature, and now is sitting in the Quick Venture Institute together with NIDCR. The aspects I want to highlight, again there are many, is value as it convey a dimension of information during COVID. Obviously, we know that of the stellar work that that saliva direct, and others has been deciphering the presence of the genomic RNA of SARS-CoV-2. We clearly is aware of that relevance and importance, but we also is aware of a piece of work that we did after we deciphered the entire salivary proteome. This is a conversation with Dr. Gil Ullman at University of Michigan. Okay, I see a hand up. Does that mean I have 5 minutes, Brittany? Okay, so I do need to rush along. So one of the outcome of comparing the saliva and plasma protein is the immunoglobulin between the two compartments in serum and also in saliva is almost linear. The message there is that if there is an isotype to an antigen in one compartment, you find it in the other. So we demonstrate that proof of concept in Sjogren, which I'll not talk about, but we sort of deploy this idea in the Sjogren, in the COVID space here. So and it was a second year dental student actually at UCLA that at the pandemic time, to obtain essential status, mask up and gown up every day, and developed the first antibody test to SARS-CoV-2 and directed against the RBD. So this ability has emerged. We were engaged in 2 RADx initiative, and this is a set of data that reflects a drop of saliva, direct detection, and utilizing 3 microliters of host saliva, 50 microliters a drop, and we see the performance of this assay here, looking for genomic RNA, antigen, antibody, and neutralizing antibody that is either at par or surpass the performance of EUA test out in the market, so to speak. So excited about this, the opportunity to do that, and being able to contribute to a dimension that certainly complement to other stellar tests out there that we know of. Again, in a single biofluid, a drop could reflect the 4th dimension of the information is important to us, and we term this Salivaomics and put it into clinical deployment. The last 3 minutes I want to share with you is this segment that we're really excited about: saliva liquid biopsy, the ability to utilize circulating targets, particularly circulating free DNA, in assessing disease phenotype in patients. So the NCI consortium has currently 6 groups that are in this particular landscape, and I want to share the work that we're excited about. This technology I'll go through very quickly. Basically, take a drop of biofluid, can release the content within the EVs, and detect it using a microarray platform or molecular ELISA that can detect target genomically, proteomically, with a performance that surpasses digital PCR and NGS. We put that to test 12 years ago. I'm looking at a year here, 12 years ago. Okay, so in looking at non-small cell lung carcinoma, these baseline sensitizing mutation EGFR, we can detect this mutation with exquisite sensitivity and performance, surpassing digital PCR as well as NGS. In plasma, we're looking at it here, but also in saliva. Our statistician actually look at this data and make the comment at the time: the saliva data, Brittany, is cleaner. It's cleaner. Okay, so take that to heart. The most exciting project perhaps we're now engaged in is with Dr. Denise Aberle, who in 2012 published a paper in New England Journal that low-dose viral CT can detect these masses in individuals' lung that beats conventional chest film by 20% in improving mortality. We put that to test, and our early data suggests that in this indeterminate pulmonary nodule, at the time of lesion detection, we can take a droplet of their blood or saliva and can identify these malignancy-associated circulating free DNA in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 55% of the IPN at the time of lesion detection, which is currently not accessible clinically, 55% having a malignant phenotype, and thereby, if it holds up, and it could be validated, it likely could allow these patients to be biopsied at the time of lesion detection. I'll just move along quickly. This is what we're excited about: a droplet of a biofluid, 50 microliter or less, of saliva can allow us to detect these circulating, tumor DNA with extraordinary performance. You can just see it. This cutoff is there, but you can see the segregation. And this platform has been mature, is clear qualified. This is the footprint, and these are the colleagues we work with in the clear certifications. And we find ourselves in this NCI consortium. What I'm excited about to all of us on the line today is that all these individuals are working with great project, advancing liquid biopsy in plasma, in urine, cerebrospinal fluid. We are there because we want to advocate saliva, and that's important. Okay, so we're in the second cycle of funding, and in that regard, the last thing I want to sort of quickly mention is that we're excited about the next generation of saliva collector, the ability to harness this biofluid and concurrently the 4 omics targets: genomic RNA, circulating DNA, and genomic DNA, and proteomics, that in one collector that could collect at the point of care and have this capability to be stored and then eventually deployed for molecular analysis in years to come. So we're working with Paul Slowy at Oasis that we exclusively use their device for our work. So on that note, I'll stop, and thank you.
Thank you very much, Dr. Wong. Lots of exciting points that I know I personally want to follow up on, but the point of this webinar is to give our network an opportunity to connect with you. So at this time, Victoria, are there any questions that came through the chat? Or if anyone wants to ask a question live, there's that raised hand feature at the bottom where you can add in a reaction, raise your hand, and once we address the questions that came through in the chat, we'll circle around and ask, give you guys a chance to ask them live. So Victoria, to you first.
Yeah, so we did have one question, Dr. Wong. If you could please share out the URL that you were referencing earlier on in your presentation.
The URL?
Yes.
Be glad to. If you can give, is it, I'm not exactly sure which one is that. So just maybe shoot me a note and I'll be glad to share that with you. Absolutely.
And that's something that we could include in the webinar follow-up
Sure.
for everybody. Thank you.
Perfect.
Hi,
Awesome.
Kitty. Judy.
Oh, yeah, I'm muted. Hi, I'm so inspired. What a wonderful, inspiring presentation. And I'm so excited about the potential of detecting quite a number of things in saliva. I did some preliminary desk research myself, and you surpassed whatever I was able to produce myself. So I'm gonna go back to reading. One of the questions that I have, and this was one of the key points from our learning from working with Saliva Direct, that we needed no stabilizing reagents in the collection tube, but and so my question is, are you doing this work without any stabilizer, or are you using any stabilizers to stabilize the RNA in the sample?
So it's a good question. It's a very insightful question, but perhaps our important learning lesson is that while all these skepticism, ourselves included, that RNA being labile could survive in saliva, the eventual discovery, if you will, that these RNA are not in free form, not in naked form. They are shielded in this EV, and thereby conferring that's integrity, stability. If you add a drop of detergent, try it next, which we did in one of our paper, they are released, and thereby just like the free and RNA and degrade rapidly. So somehow these bodily constituents are encapsulated and thereby conferring that stability.
That is super interesting. Thank you.
It is.
We have time for one more question if anyone else would like to ask something of Dr. Wong before we move on to the next presentation.
We do have one in the chat, Brittany. This is, could you comment please on the relative utility of sputum versus saliva? And this is for infections and cancers.
So I think we're talking about two adjacent, perhaps bodily fluid. Sputum is sputum, and saliva is oral fluid, and perhaps do they overlap? I'm almost certain they do. But I guess in the context of this webinar, perhaps if you will, I think our task is to really foundation and assess and project the clinical utility, and perhaps more importantly, the scientific foundations. So yeah, I think that's perhaps is a way to address that.
Thank you for that. All right, if there are any other questions that come through the chat, if we've got time at the end, we'll circle back around to them, and we'll certainly follow up with you, Dr. Wong, to get those URLs so we can share them out in the follow-up presentation.
Will do.
Thank you. All right, now we're happy to transition over to our next speaker, Professor Chamindie Punyadeera, who joins us from the Griffith University in Australia, and we are very appreciative. She's waking up very early to join us today. She is a professor and the head of the Saliva and Liquid Biopsy Translational Laboratory, and her presentation today is Disease Detection is Spitting Distance Away.
Good morning, all. Brittany, can you see my slides?
Yes, we can see it. It's not in presentation mode, but we can see your slides. Now it is.
Can you see? This is all good, right?
Yes.
Good morning, all. If you're from the US, I think it's good afternoon, or anywhere in the world, good day. Thanks, Brittany and Saliva Direct for giving me the opportunity to present some of the work that's happening in my lab. This is my disclosure statement, funding and commercial success. Today I'll be showing you work relating to various aspects where we have used saliva to early detect cancer, and in particular head and neck cancer, lung and glioblastoma or brain tumors, heart failure, because there is now very clear evidence, as Dr. Wong demonstrated, that systemic biomarkers land in saliva, because they've beautifully shown in, I think, a paper about a decade ago where biomolecular transportation occurs, and we can use saliva as an adjunct body fluid to early detect systemic events. The research program that I'm leading covers multiple aspects. So we predominantly working on cancer detection, and we are also using bugs in the mouth, and in association with, specifically for the development of oral and throat cancers, early detection of heart failure, as well as liver fibrosis using saliva. 3 major programs. So we do biomarker discovery. So we bring biomarkers from concept through to commercialization, and I'll demonstrate 2 successful commercial products from my team. We do bioassay development. It's not easy to use saliva and to develop assays to detect microRNA or proteins because of viscoelastic properties of saliva. And we are also working with engineers, very clever engineers. And in fact, we just finished an NIH grant with Prof. Chuck Henry, sorry for that spelling, from Colorado State University, where we've developed a paper-based analytical device to detect biomarkers, in this case, proteins present in saliva, and particularly for heart failure. And we are also developing a wearable diagnostics where we are trying to detect real-time proteins and microRNA using a saliva denture. We work on liquid biopsy space, and that's particularly using blood as a diagnostic medium. I wouldn't be showing you this work today in this talk. Head and neck cancers is the 7th most common cancer. There are about 1.1 million new cases worldwide, with 500,000 deaths. The 2 major types of head and neck cancers are oral cavity cancers caused by smoking and alcohol consumption, and oropharyngeal cancers caused by human papillomavirus. Just like the cervical cancer that's caused by HPV, when HPV lands in the throat area, it gives rise to oropharyngeal cancers. Sadly, most patients die within 5 years 5-year survival is about 50% because of late diagnosis. And 30% of patients develop recurrences. So we really need tools to early detect and to monitor response in head and neck cancer patients. This is a work we're really proud of. This work started about 8 years ago, where we were discussing with Wong. That time, Wong was a health and wellness company, and I asked them to use saliva, especially the bacterial gene expression in saliva, as a biomarker panel to early detect oral and throat cancers. And in 2022, Biome received the FDA approval under the breakthrough device designation for cancer detect, which is very, very simple. I'll just show you. I have a video here and I hope it will play. So that's a box that comes to you and it's about $399 US. And the box contains saliva collection with the correct stabilizers where you can FedEx this box back to Biome, and where bacterial RNA is isolated and done metatranscriptomics on next-gen sequencing. So we've developed an AI and machine learning tool, and we've developed a biomarker panel that can accurately detect oral cancer with a sensitivity of 90% and throat cancer with a sensitivity of 90%, and specificity is about 85. And that's Dr. Guru, the Chief Scientific Officer of Biome. So the second product is a companion diagnostics for heart failure. Heart failure is the inability of the heart to pump sufficient blood to the flow. And then what happens? You get a situation like a flying donkey. Heart failure is a global pandemic affecting over 64 million people worldwide. What we did is that we used saliva collected from age-matched healthy controls, early phase heart failure patients and late-stage heart failure patients. We did a mass spectrometry to discover novel proteins. Then it went route of validations and again validation, and we identified a biomarker panel, 6 biomarkers, as you can see, the sensitivities and specificities, which was licensed to a startup company from Australia called ESN Clear. We are using saliva as an adjunct or a diagnostic medium to early detect HPV-driven oropharyngeal cancers. And if you guys know, you all know Michael Douglas. And a couple of years ago, he came out saying he's diagnosed with oropharyngeal cancers, and that's due to oral sex. The way the workflow goes is we have saliva, so we do an oral swish and gargle. The reason why we are using a swish and gargle is to get the cells that are infected with virus, because the virus resides in the throat area. Samples are brought to my lab. We do DNA isolation, and we have developed a 4-plex high-risk HPV panel that covers over 98% of high-risk HPV types to detect within 2 hours. And this is done on both the digital PCR and qPCR. This work started before COVID when Johnson & Johnson, they were developing a therapeutic vaccine. We all know the Gardasil vaccine is a prophylactic vaccine, so it can't protect people who are already exposed to HPV. And Johnson & Johnson, back that time, they were interested in developing a companion diagnostics for their vaccine. And as part of that, we recruited 665 cancer-free individuals from University of Queensland Dental School. And those days, we only had one assay going, that is for HPV-16 DNA. And we detected in 12 people, 12 out of 650 people, HPV. So it gives a prevalence in Brisbane about 1.8. 3 of them were persistently positive for oral HPV. We know that from cervical cancer literature that persistent people are at risk of developing head and neck associated cancers, especially oropharyngeal cancers. We got them back into the clinic, and Associate Professor Saj Vasani is the ENT surgeon who saw these patients. The first patient had no cancer. The second one was a woman. She had pre-malignant lesions, and then she told us that she was also diagnosed with cervical cancer. The third one, it's the first time that we were able to detect a 2-millimeter occult cancer at the back of a throat of a healthy person. And he was a retired teacher who was English teacher working in Bangkok. And when he was seen by Saj, he had HPV DNA viral copy numbers about 1,200. And he was then sent to get an MRI. MRI came negative, but Saj found the left tonsils to be swollen. But that doesn't tell you that you have cancer. But the patient opted to undergo tonsillectomy, and it's the first time that we were able to detect a 2-millimeter cancer in a healthy asymptomatic person. And 2 weeks later, he was then again seen by Saj post-operatively, and he's had no HPV. So literally, we've closed the loop that HPV can be used as a diagnostic as well as a disease surveillance tool. And this work was also backed by a study from Hopkins saying that they were also able to detect occult cancer in a healthy person. So we were very much interested, if we can detect HPV-16, what's the correlation between HPV-16 and the current gold standard, which is the tumor P16 expression. So we recruited 268 patients, and we collected saliva and we tested saliva for HPV DNA. We found high-risk HPV types in 72% of the cohort. Majority of them had HPV-16. And you can see that most of the patients who were HPV positive had tonsillar or base of the tongue cancer. And in this table, what we did is that we tested for HPV, and the concordance between, or the correlation between tumor P16 and HPV is about 99%. And we can also use HPV to determine cancer-specific survival. In this figure, you'll see just solely using high-risk HPV, people with high-risk HPV positive do well. Those who are HPV negative, the red color one, don't do well. And if you look at P16, similar. However, if you combine HPV and a tumor P16, the blue line, you get a better prognostic power. MicroRNAs are quite stable, and we now know that microRNAs are non-coding, but they regulate gene expression. And we did sequencing in saliva samples collected from a cohort of HPV positive patients and HPV positive control. So we are comparing cancer versus control. Underlining biology is HPV. So any difference that we see in microRNA gene expression should be caused by cancer. And this is in the validation phase where we have oropharyngeal cancer patients, positive and negative. So if you look at OPC, the purple ones, and the controls who are positive, the red one, you can see the median values increase for most of this panel. And we identified 3 markers with high sensitivity and specificity. So we can use these microRNAs to early detect HPV-driven oropharyngeal cancers, which is currently an unmet clinical need. About 20 to 30% of these patients develop recurrences, sadly, within 5 years, because we can't pick these cancers early. Early diagnosis of oral cancers. So we are using a similar platform as the oropharyngeal, like microRNA gene expression. And this is John Farnham, Australian singer. He was diagnosed with oral cancer in 2022. Sadly, he even had to undergo jaw reconstruction. Oral cancer is the 16th most common cancers. 5-year survival is 50%. And we are looking at transition from pre-malignant state or oral potentially malignant disorders to oral cancer. And currently, there is no screening program to early detect oral cancer or to predict the development of oral cancer in pre-malignant lesion patients. So we did a sequencing, we did validations, and then we identified 8 microRNAs that are differentially expressed between controls, OPMD patients, and oral cancer patients. And you can see that in the healthy controls, light blue, oral cancer, dark blue, for MIA7, it increases. And then we developed a sophisticated algorithm based on AI and machine learning to develop a diagnostic algorithm. If you look at the tissue samples, for MIA7, we've only demonstrated for MIA7, you can see that MIA7 in the tissue is elevated compared to a healthy oral tissue, also for 10-5B. So for most of these samples, tissue expression coincide with saliva expression. And this shows the early detection ability of salivary microRNA with the area under the curve of 0.96. If it's 1, that is 100% sensitive and 100% specific. And to discriminate pre-malignant lesions from oral cancer. And you can see it's quite clearly in a larger cohort, we can discriminate these patients, which tells us that we can use these microRNA also to predict the development of oral cancer in OPMD patients. I see Brittany's hand. I just have to skip a lot of my work then. So when we did this work, we found that in the low-grade OPMD versus high-grade OPMD, microRNA expression changes increases, as well as in cancer. 2 of the patients at the time of analysis, we found that these patients had elevated microRNA expression, quite similar to cancer, and 6 weeks down, they were diagnosed with cancer. This clearly demonstrate that microRNA gene expression can predict the development of oral cancer. I'm now going to have to quickly go over the use of saliva to discriminate benign versus malignant lung nodules using DNA methylation. I'm not putting these genes because we are in the process of filing IP. And you can see that in the benign group, the green color group, and the malignant group, the DNA methylation changes differ. And these are the sensitivities and specificity. And using cell-free DNA in plasma, similar results. We also used exosomes, and you heard from Dr. Wong about the importance of exosomes. These are the key messengers. And we were able to use exosome as biomarkers to predict outcomes in patients with GBM. And we identified the protein cargo in GBM patients. And you can see that when you do the validation, ALDO-A1, which is an oncogene, is increased in patients with recurrence versus patient without recurrences. We've also used saliva to early detect liver fibrosis, because over half of the adults populations is diagnosis with liver fibrosis, which then progress to liver cirrhosis, then to liver cancer. And these are the markers that's already currently used for diagnostic purposes. And what we did is that, are these markers present in saliva? And as you can see, this is the serum and salivary levels. And yes, there is also correlation, and we can detect most of these markers in saliva. So how would the world be? I think saliva would play a major role in disease diagnosis, screening, risk assessment, as well as especially in cancer, monitoring response to treatment or disease surveillance. With that, I would like to thank my team and the funders, and happy to take any questions you have.
Thank you, Dr. Punyadeera. Appreciate it. Thank you for that. If anyone would like to use the raised hand feature, we're happy to give you permissions to be able to ask a question verbally of either of our speakers. Since we have a little bit of extra time, Dr. Punyadeera, you mentioned having to rush through some of your research. Is there anything that you were really wanting to touch on that you didn't have the opportunity to, since we've got a couple of minutes?
Yes, I just didn't get to go through over the, I think I go over the background. But what I'm trying to point out here is that when I started this program about a decade ago, people were very skeptical. How can you use saliva to detect heart failure, cardiovascular? What's the connection between saliva and blood system? But lo and behold, we didn't understand then how these different molecules get transported across. And we now kind of know that there are different mechanisms like diffusion, active transport, and through the gingival crevical fluid, these biomolecule So I think saliva is a treasure trove. You can use saliva for a multitude of diseases, including systemic diseases. So that's a point I want to make.
Absolutely. That's actually one that I'd taken out of both of your presentation as well as Dr. Wong's, that point that a lot of your work started with skepticism, or just skepticism of what we could learn from saliva. Curious if you or Dr. Wong, feel free to tag in, could speak to what you think has caused a transition or a shift away from that skepticism. What's really helped demonstrate its usefulness? Is it just the science, or people are getting more comfortable with the idea of that, like as saliva as a sample type, or what do you think is helping?
So there have been studies linking, like David mentioned, right? Studies clearly demonstrating that systemic diseases changes can be detected in saliva. So that's one important aspect, the scientific foundation. And the other aspect is that I always call the positive side of COVID-19 is that everyone understood the importance of saliva testing and importance of non-invasive testing. Even my dad, who is 84 years old, understand now a PCR assay is more sensitive compared to an antigen assay. He doesn't understand what does it mean, the underlying biology or the principles, but whenever I get a cold or fever, he says, you can't do the antigen test that the Australian government distributed freely. You've got to get the PCR and stand in the queue. So there you go.
Thank you. Dr. Wong, anything you wanted to add? Related to this, how we transition from folks being more skeptical over to more accepting of saliva. Yeah.
Well, the skepticism is good. Perhaps what I think the lesson that's taught there is we just need to do the science. And so because you talk to someone that's a 96-year-old, 40-year-old and say you could evaluate the disease through saliva, everyone gets it. Everyone gets it. But to this day, I mean, the number of saliva tests or evaluation to traverse through regulatory approval is single digit. So sure. It tells you that, while the sort of the opportunities, if you will, is abundantly there, it's still the scientific foundation in particular, the ability to definitively evaluate these constituents for disease detection. It's here. We know how to, but perhaps just having that discipline and perhaps that stubbornness to go through the quest itself. But yeah.
Thank you. I think that's a really nice note to close out on, even, and something that makes me very grateful for the work that both of you continue to do. Makes me very proud of the work that our team at Saliva Direct does, and also so many folks that are on this call and in this network, and what we're continuing to push forward the fact that we even have all of these speakers and great interest in saliva diagnostics, I think is a great indicator of where we're heading and where we are now. So really appreciate both of you speaking with us today. We hope that we'll get to see many of you at future webinars that we're going to be hosting. Please feel free to send in your suggestions. Again, thank you, Dr. Wong, and thank you, Dr. Punyadeera, for joining us today. Really appreciate you guys taking the time and sharing your expertise with our network.
You.
Thanks a lot. Bye.
So,